Welcome to the first video in our series covering Mendelian genetics. The purpose of this video is to review the key concepts of the cell cycle, which is the complex biological processes that your cells use to decide whether they are to reproduce or continue going about their day-to-day functions. Before we talk about the details of the cell cycle, we need to look at how human DNA is organized within a cell. So unlike simpler organisms like bacteria, your DNA actually does not exist as a single piece. It exists as 46 separate fragments, which are what we call your chromosomes. Now, most biologists don't actually use the number 46. They find it more useful to describe your chromosomes as two sets of 23 chromosomes, even if these numbers are identical, because this tells us a little bit of information about where each of these chromosomes came from. Now, half of your DNA, from common sense, we know that you get from your father. So 23 of these chromosomes are collectively what we call your paternal chromosomes, and the other half, the other 23 chromosomes that you get from your mother are collectively what we call your maternal chromosomes. And when we combine both your paternal and maternal chromosomes in a diagram like this collectively is what we call your karyotype, which is a record of all of the DNA that exists in all of the cells of your body that actually contain DNA. Now, karyotypes are a little bit misleading because a lot of people think that whenever you look at a cell, you can count 46 different pieces of DNA that are visible, but your chromosomes actually only form these structures right before the cell is about to divide into a daughter cell, meaning that you actually never see these structures most of the time unless a cell is in the process of reproducing. The rest of this video is going to look at how the control center of your cells, what we call the nucleus, regulates the process of deciding whether a cell is going to perform its ordinary day-to-day functions or whether it needs to reproduce. And, well, what I just said, those are the two basic things that every cell can do. Cells can grow, they can get bigger, they can mature, and they can perform their daily functions, or they can decide to make a copy of themselves, which is what we call cell reproduction. The second video in this series covering the process of mitosis will go over this in more detail. So before we talk about this process, we need to actually look at the conditions that tell a cell when should it be performing its day-to-day functions and when does it need to reproduce. Well, normally most cells in the body are not actually reproducing. Most cells are actually undergoing their normal day-to-day process. So most cells are not actually dividing. Most of them are performing their regular functions, whatever the regular functions is. So if this is a brain cell, its job is to communicate with other cells in your body. If this is a white blood cell, its job is to float through your blood and hunt down anything that doesn't belong in your body that can make you sick. When cells are doing their regular functions, they are said to be outside of the cell cycle, which is defined as what we call G0. However, should an environmental condition that comes in necessitate cells reproducing. So one simple example is if you cut yourself, that means that you've killed off a small amount of your skin cells and your body will need to make more to cover up the wound and help you stop the bleeding. Now, in order to do this, you would need to take a cell from G0 and cause it to enter the cell cycle so that that cell can reproduce and turn into two cells. And when a cell moves out of G0 and commits to undergoing reproduction, this is what we call the G1 phase, also known as the gap 1 phase, although many biologists will refer to this as the growth 1 or first growth phase, because these cells, if you want to take one cell and you want to turn it into two, these cells need to grow bigger so that you have double the amount of material that is necessary in order to make two cells. Now, obviously, this would require a lot of energy to be able to do, and this would also require all of the organelles, all of the individual components, such as the mitochondria, the endoplasmic reticulum, the Golgi complex. All of these would need to duplicate itself in order for cell division to occur, and all of this occurs during the G1 phase, which is why this actually makes up the largest stage of the cell cycle. Now, one thing that you'll notice that is not copied or produced during the G1 phase is the cell's DNA. In fact, there is an entire phase specifically devoted to the duplication, that being making 2 copies of your DNA, and this is what we call the S phase that occurs after G1, which is what we call the synthesis phase. And again, the job of the synthesis phase is to take all of the DNA that exists in your cell, so the 2 sets of 23 chromosomes that you have, and double them so that you have enough DNA not just for a single cell, but for a second cell that we are going to make at the end. Now, the problem with DNA duplication is a lot like the problem that all humans are prone to. Let me give an analogy. Let's say that you are working on an essay or a short story, and rather than deciding to just hit Control-C and Control-V to make a duplicate of it, let's say that you want to be extra careful and you want to actually type out a second identical copy of whatever it is that you've written. Now, how many people do you think can actually read out their original copy and type it completely perfectly without a single spelling, grammatical, or syntax error? I'm guessing that no human is able to do this with 100% certainty. Call it human error, if you will. And your cells are actually no different. Your cells, when they duplicate their DNA, are going to make mistakes. In fact, it happens quite regularly, but that's why we have the G2 or the gap 2 phase. Because what do you do whenever you need to write something before you submit it? Chances are you would proofread whatever it is that you had written in order to catch as many of the mistakes that you make simply due to human error. And that's exactly what the most important stage of G2 is to proofread the DNA that was duplicated in the S phase. Once this proofreading is done, the cell is technically ready to undergo cell division. Only we're missing one thing. A large number of proteins, such as enzymes, your cell's biological catalysts, are necessary in order for mitosis, the process of cell division, in order to occur. And once the DNA produced in S phase has been proofread, and these proteins that are necessary for cell division to occur, the cell has passed what we call the G2 checkpoint and is ready to actually start duplicating itself in the process that we call mitosis, which is more commonly known as cell division, where we take a single cell, what we call the parent cell, and where we take all of the material in that cell, so all of the DNA, all of the organelles, the mitochondria, the endoplasmic reticulum, even the nucleus itself, as well as the liquid part of the cell, what we call the cytoplasm, and where we divide that from one parent cell into 2 daughter cells, such as the ones that we see here. And if this is done completely perfectly, each of the daughter cells will have identical DNA to the original parent cell that acted as the blueprint in order to make the second cell. And once these 2 cells have been produced, assuming they are ready to perform their normal functions, they would exit the cell cycle and enter back into G0, where they would continue performing their regular function until it was necessary for them to divide once again. Now, the process of regulation is a lot more complicated than this. Anyone who studies biology in university will learn a lot more in detail, and one of the things that they'll learn in more detail is what happens when this goes wrong. So, the cell cycle is very important to regulate because its problems with cell cycle regulation result in cells that can divide too often, or cells that don't divide at all. And now both of these are very, very, very problematic, because if a cell can't divide at all, then your body is not able to replenish cells when it gets injured or when it gets sick. But when your body has cells that divide too often uncontrollably, this is actually a very well-known disease. This is actually the fundamental basis of cancer, when your cells lose control of their cell cycle and the cells are effectively stuck in an infinite loop of the cell cycle itself. And because cells that are in the cell cycle require much more energy and nutrients, cancer literally starves your body to death, producing cells that it actually doesn't need. In the next video in this series, we're going to take a detailed look at the stages of mitosis to look at how the cell actually turns from a single parent cell into a pair of daughter cells.